So you have so that's all right. I can just. No, but the thing is, once it's blocked, Oh, 1 second. Okay. Now it works. This doesn't work. Okay, so I think so we can start. First thing, David, thank you very much for being here and also to your research group. So I met David already many years ago when I was doing the PhD thesis in Santiago with Samuel Emilio, and you came to visit us. It was already 15 years ago, probably, or something like that, or even longer. So David has a very interesting background because he mixed a bit of science and culture and also, I don't know, a lot of things in his research. So he did a master in cultural theory in 2004 in the University of Manchester, and then he did the doctorate in physics at the University of Leeds. I think you graduated in 2008. Right? And after the PhD, he received a lot of prizes, like the Royal Society Research Fellowship, the ERC starting grant or consolidator grant, which one? The consolidator grant. And he published in a lot of papers in many research fields in computational chemistry, but inside computational chemistry, he worked in classical dynamics, quantum dynamics, in also biochemistry, computational biochemistry. He developed a lot of algorithms. So in the last years, he was also working in these virtual reality tools, and we will learn today and tomorrow also in the demos. And yeah, he's a very versatile research, and I like very much to do research. So thank you for being here, and we are looking forward to hear your presentation. Thank you, David. Yeah.
I just, if I speak here, it's okay? They can hear?
Yes, okay.
Okay. All right.
The microphone is here, so maybe you should
okay.
A bit louder.
Okay.
It's okay. It's okay.
Okay. All right. Well, thank you very much, Juanjo, for the introduction, and thank you to everybody here. And so thank you to some members of the research group from Santiago have come to show some demonstrations of the virtual reality and run some experiments over the next days. And so I'll talk a little bit more about the presentations that we will hear from the other members of the group later in the talk. But we have a laboratory in Santiago de Compostela, which should be called the Intangible Realities Laboratory, so the IRL. So we have, we're supported by a few different funds from the ERC and the American Foundation, the Tiny Blue Dot Foundation, the Junta de Galicia. And so this is a picture from previous version of the lab. But you see there's two researchers that are wearing virtual reality, and this is actually two of my friends who work in psychedelic neuroscience, and they're inspecting a crystal structure of the human HT2B serotonin receptor, which is a brain receptor. And what I like about this image is that you can see that they're both looking at something which is approximately here. Right? And this is real for them, but we cannot see what they're interacting. Right? So this is why we call the lab the intangible realities laboratory, because for these two researchers, this is a tangible reality, but for people that are outside of the virtual environment, it's an intangible reality. So that's why we have that name for the laboratory. And so in this short talk, this introduction, I'm just going to give a very, very brief background to molecular visualization and dynamics, like very short history. And then I'll talk about a technology that we've been developing in the laboratory called interactive molecular dynamics in virtual reality, which we abbreviate with this acronym, iMD-VR. And then I'll talk about some applications, and I'll specifically talk about some applications in quantum chemistry and then in interactive finding. So yeah. So I just want to start with a very quick history of virtual reality. And so the first ever virtual reality system was pioneered by a researcher at Harvard in the 60s called Ivan Sutherland. And he wrote a really famous paper called The Ultimate Display in 1965, which is only 2 pages long. So it's very easy to read this paper. And it would be interesting for everybody to look at this paper if you're interested in this area. But this is the very early 1960s version of virtual reality in the lab at Harvard. And if you read this paper, what he says is really interesting is what he was thinking about was physics application. He wasn't thinking about video games, and he wasn't thinking about watching movies. He was thinking about how virtual reality could be used to understand physics in a better way. And so, what he writes is, he says, we live in a physical world whose properties we have come to know well through long familiarity, which gives us the ability to predict its properties well. For example, we can predict where objects will fall, how well-known shapes look from other angles, and how much force is required to push objects against friction. We lack corresponding familiarity with the forces on charged particles, forces in non-uniform fields, and high inertia, low friction motion. And so, what's really interesting about this quote is, what he says we don't have familiarity with is exactly the regime of quantum physics. Right? So he doesn't say anything about atoms and molecules here, but forces on charged particles, forces in non-uniform fields, and depending on the density, high inertia, low friction motion are all very specific domains where we care about molecular physics. And so, a lot of the work that I'll talk about over the next 20 minutes is work we've done to understand molecular physics in this regime here. So I'm going to give a very short history of molecular visualization and dynamics. I'm going to miss out a lot of things. But basically, this is conventionally how most people think about molecules. A lot of people imagine molecules as rigid structures. So maybe if you study chemistry at school, or high school, university, you've used these kinds of models of little atoms connected by sticks. So, this is a small molecule. This is a bigger molecule. This is Watson and Crick, who, were awarded the Nobel Prize for the discovery of DNA. And here they're using a much larger model, but it's the same idea as a small model, right? It's a rigid model, and it does, and it tells you something about structure, but it's not dynamic. So this is some work we've done with virtual reality to visualize static structures, so rigid structures that do not move. And so earlier I showed you this image. So now I'm showing you what those two researchers are looking at through virtual reality. Okay. And so this is a cell membrane. This is a slice of cell membrane with the human HT2B serotonin receptor. And what we're looking at, what the two researchers are looking at is they're going into the cell membrane and they're actually looking at the crystal structure from the protein crystal database. And they're looking at the structure of the LSD molecule, which is bound to the HT2B human serotonin receptor. And this is one of my colleagues, Professor Robin Carhart Harris. Show you this one more time. So he's a psychiatrist and a neuroscientist. And I remember the first time that I showed him the system, how excited he was, because he had only ever studied this system sort of at a very macroscopic level. He had never had the opportunity to actually see the crystal structure of what this system looks like. And so he was really excited when I was able to show him the structure of LSD bound to this receptor. And so this is an example of how virtual reality can be used to visualize sort of rigid structures that do not move. So it's cool and it's cheaper and it doesn't take up a whole room and you get more atomic detail. And you can visualize, for one of these models to make a 3D model takes a lot of time, money. But to visualize a model of virtual reality is much easier. You still get the sort of same three-dimensional impression as a physical model. But molecules are not static, they move, right? And so life arises from the dynamics of molecules, molecular dynamics. And so this is a paper that I wrote a few years ago, which is a simulation of diamond. So this is a real-time simulation of diamond. And this is often a surprising movie to people because diamond is one of the hardest that we know of. And so most people would imagine that if something's hard, it does not move. But it turns out that part of the reason that diamond is so hard is because of the way that it vibrates. Right? Actually, it's vibration what make it so strong. And so I like to show this video because this is often surprising to people if they would guess that something like diamond not move, but it does move, and it moves in this really sort of beautifully choreographed way. And if you know something about solid state physics, you know that the way that it vibrates are often called phonons, the vibration of the material. And so, Richard Feynman has this very famous quotation where he says that everything that living things do can be understood in terms of the jigglings and the wigglings of atoms. I like this quotation because it emphasizes this point that nature is dynamic. And so if you only think about molecular structure without thinking about motion, then you miss an important part of the mechanics of how living systems and even non-living systems work. And so there's a whole field of molecular dynamics, which Wanho knows about and works in. And it's a massive field. And there's been a number of people across the world over the last 50 years that have worked in this field. And it goes all the way from very small molecules, this is OH plus methane, and to much larger molecules. Sorry. This is HIV protease. So this is one of the molecules that's targeted in some HIV therapies. It's a molecule called Methodological. So it's HIV protease. And so this is showing how the small molecule binds the binding pocket of the protein. And so, yeah, there's a lot of people working on the simulation and visualization of the motion of molecules to understand, for example, structural changes in proteins or how chemical reactions out of atoms for between different molecules. And so, the simulation of molecular science kind of started in about the 1970s, was when a lot of the original work was done with the first ever molecular dynamic simulations. And so since that time, computers have gotten more and more powerful, and simulations have gotten better and better, and graphics consoles have gotten better and better. And so, if you kind of imagine the arc of simulations and molecular modeling, it's something like this. Here's a time when you have Watson and Crick building sort of full room scale models of proteins, DNA, and now most people sit at their console and do molecular modeling in this way, right? And the way that most simulation works now is that, individual agents, people submit batch jobs to remote supercomputers. So usually if you're doing a molecular simulation, you're not working on your own computer, you're communicating with a bigger computer, you're just using this as a console for the larger computer. And I guess one of the questions that we've been that's motivated a lot of the work in virtual reality is this really the best way to approach scientific molecular simulation, right? Maybe that there's certainly a role for this, but maybe there's other ways that we use our bodies, so that we're not sitting at the desk all the time, to explore molecular systems in different ways. And maybe that helps because a lot of people that study molecular science have developed back pain after years. So maybe if we have other ways of doing molecular simulation, we have a little bit less back pain, right? It's a little bit more fun. So the problem with the reason people use big supercomputers and the problem with other approaches is that molecular simulation requires very complicated physics. Because of that, it's typically only possible to simulate very short timescales unless you have a really big computer. So people need big computers. And in order to get meaningful results for most molecular simulations, you have to use a technique called biasing. So you have to push the simulation somehow into the area that you're in, the region of space that you're interested in order to encourage it to undergo the structural transformation of it. So if you know something about the system, you can prepare your simulation in a way that helps the system go into the area of interest. And there's a whole field of how to bias physics simulations. And I'm not going to go into that today. I'm just going to talk about the method we've been developing virtual reality. And Harry and talk more about this in his talk, for example, and so on. Okay, so when we bias, one way we can bias simulations is we can use interactive molecular dynamics in virtual reality. So this shows biasing of an OH molecule to have it do reactions with a methane molecule. So if I take the OH, I can literally take molecule and I can simulate a collision. So this is being run with a density functional theory model. This is called tight binding DFT. And so what the researcher is doing is taking the OH and guiding it toward the methane. And what's really interesting is if you watch this video, you see a whole bunch of different kinds of collisions. So that's a reactive collision. You'll see some non-reactive collisions. So that's another reactive collision. You'll see what's called a chattering simulation, where the hydrogen actually transfers over from again. You'll see one reaction where the hydrogen actually goes over to the OH and then it goes back to the methane. Let's see. And you'll see some collisions that are direct collisions, and you'll see some collisions that are more sort of indirect collisions, but are still reactive. And so this is a way of doing biasing. And the other thing you'll notice is that when the water molecule is formed, it has vibrational excitation. That was the chattering collision. You see the hydrogen temporarily transferred to the OH and then it went back. And interestingly, that's been experimentally observed. So that was a really cool thing that came out of this was, one student throwing OH at water was able to observe all the class simulation of collision, reactive and non-reactive, and study experiments. It's very cool to be able to do that in 10 minutes. You actually can gain real molecular insight. And so we published a paper back in 2018, where we kind of outlined this new way of doing molecular simulation. Using virtual reality with real-time physics and cloud computing. And so we published this paper and it generated a lot of interest from people. And we outlined this paradigm we call iMD-VR, interactive molecular dynamics and virtual reality. So this shows one of the schemes in one of our papers. This is two researchers in the same space playing with a Buckminster fullerene. And here's actual video that we made in the lab. This is two of the PhD students that were in the lab at the time, Mike and Becca. They're tossing the Buckminster fullerene molecules back and forth. And I guess the thing to emphasize here is normally when you're running virtual reality, you use the physics that's available in video game engines. But we're not doing that here, right? We can't use video game physics because video game physics is typically rigid body physics. And we know that molecules are not rigid bodies, right? They're flexible and they move according to electrostatic forces from van der Waals fields. So we wrote, in order to make this work possible, one of the things we had to do is write our own engine. Well, we use existing engines, but we had to write an interface to existing molecular simulation engines to allow the physics that's being simulated when you touch a molecule to be real physics, right? So this is the real physics of simulations of the things are passing the molecules back and forth. And so the way that this works is pretty simple. These are the only equations that I have from the talk. So there's a lot of ways of simulating molecular motion, but they all basically have the same idea, is that you update your position and your velocity to go from your current time to some future time, delta t. And to do that, you need to know what the force is on each atom time t, right? And so you see that the force term occurs in both the position update and the velocity update. And the computational cost of molecular physics is evaluating this force. So that's where you pay the price, right? So what we do is, so the force term is basically the negative derivative of the potential energy, which depends on all the coordinates, q. So there's 3n different coordinates. And so what we do is we split the potential energy up into two terms. We have an internal contribution of the potential energy and external contribution of the potential energy. And then that lets us write the force as a contribution from two terms, the internal contributions to the force and the external contributions to the force. And so the internal contributions to the force are what you get from any of your favorite molecular physics simulation frameworks. It could be molecular mechanics, it could be ab initio, it could be semi-empirical, it could be fancy functional theory, whatever. And we've experimented with many different ones over the years. And then the external force is what the user is doing to the system. So when I reach into the system and touch an atom, I exert a force on it that's integrated smoothly into the equations of motion. And that's what lets me guide the motion of the system in a fluid, physically rigorous way. And we outline how, if you're interested in this, you can read this article. This article explains how to do it. And in order to be able to do this in real time, we use GPUs, right? Because GPUs allow you to do computation faster. So people here have probably heard of GPUs. They're being used. They're almost impossible to buy now because everybody's using them for artificial intelligence. But the basic idea of a GPU is that instead of a CPU, which has, a few cores, which are very versatile, do many different kinds of logic, GPUs have, thousands, even tens of thousands of cores, but they're kind of stupid. They can only do very basic floating point arithmetic. But what a lot of but if you can rewrite your problem in a way where you can just split it into a bunch of sort of atomistic math operations, then you can use these tens of thousands of cores to do really sophisticated computation very quickly. So we use GPUs when we do the simulation, and that's what allows us to do things in real time. And so when we published this paper, it made a lot of people excited. So it got it seemed to captivate people's imagination, and we got covered in the New York Times, which was very cool. And the researchers highlighted how if they had a tool for learning chemistry this way, maybe they would have been much more excited about this. Because a lot of students find chemistry kind of boring in high school. So here's a few examples of systems that we've published. This is interactively biasing a protein to tie it into a small knot. This is like a nano knot. And so there's a number of structures that occur in nature, and even recently in antibiotic structures that form knots. And so this is an area of interest. And then this is another system where we're taking a methane and we're threading it through a carbon nanotube. And I think Mohammed will probably talk more about knots in the talk that he will give. And this is another thing we've done. We've been looking at new interfaces for manipulating molecules. And this is a glove that we published a while back, which you could wear. And instead of using the controllers to manipulate a simulation, we could just manipulate the molecule by touching your fingers together. Now there's been advancements in hand tracking, which allow you to do this without wearing the glove. So when people see Raz's experiment tomorrow, you'll have the ability to touch the molecule without wearing a glove, just using your hands, which is tradition. And so we published a lot over the year of the quantifying the benefits of VR, so one of the questions that emerged when we started doing this work and still emerges is, well, I have all these 2D ways of doing chemistry. Why should I use virtual reality? And so we've done a lot of work to quantify why. And I won't go through all of it, but we've shown in a range of papers that when you study molecular objects in VR, and you compare in 3D, and you compare it to how you do things in 2D, you typically get accelerations in the rate that you can study systems that's 2 to 10 times faster. And it's interesting, there's been some studies in computer animation, actually for studios that develop like movies, like Pixar, where they've had a huge effort where their animators don't use 2D anymore to make, track or whatever. They use 3D, they use VR to do it. And they've shown very similar accelerations, about 10x faster to do normal things, because you have 3 dimensions. So that's kind of obvious. When we published this, a lot of people were surprised. I wasn't surprised, but it was important to do this to show people that, it's faster. And this was kind of a cool paper we published in 2019, where we showed that if you use neural nets to train, to learn solutions, to learn potential energy surfaces, solutions to the Schrodinger equation over large regions of configuration space, that the neural nets learned about 10 times faster if the data was sampled in virtual reality versus standard sort of data gathering approaches. And that's just because you get higher quality data faster. And so neural nets like better data, they learn faster when they have better data. And so that was kind of an interesting application. And then there's also a number of qualitative benefits of iMD-VR. This was a paper we had in the Journal of Chemical Education a few years ago, which showed the benefits of using this as a teaching tool. So specifically looking at computational enzymology, and showed that students really liked it and they got better grades. So again, that wasn't so surprising to me because a lot of research in psychology and neuroscience shows that our attention is enhanced when we engage in multi-sensory processing. Right. So, if you're studying things just using your eyes all the time or writing, you're not using like other things that your human organism can do, like movement and touch and vision and integrating with proprioception. And so, of course, it's better for education. And the students remember things better because they're engaging more of those senses. And we've also shown that, it enables more efficient communication between scientists and teachers and students to accelerate molecular research and learning. And that's just because you have another dimension. So, right. So I'm just going to go through a few examples and then I'll finish of stuff we've done with research over the years. So one thing we've done is we've used iMD-VR to explore kinetic networks on reactive potential energy surfaces. And by the way, I'm going to say Harry and Mohammed and Glowacki are going to talk about other applications. So I'm just going to cover some applications that they probably won't talk about. And then I'll let them talk about the rest. But yeah, so when we study and Bonho knows about this stuff, but when you study the dynamics of molecules, you're often interested in what reactions they can undergo. What if I have OH plus this thing here, propine, what are all the possible reactions that can happen? And this is called mapping the potential energy. And it's often really complicated because there's lots of structures and lots of things that can happen. And so that makes it hard. So one of the areas that we've looked at over the years was this was work by Robin Shannon in 2021. Can we develop like a game where then master students could come and play the game and help us solve all the structures that are possible on a potential energy surface. So we did this by coupling the iMD-VR framework to density functional theory. And so you're going to see a student playing the game, looking at all the possible reactions. And then, when a reaction happens, they get a notification and it tells them that there's a reaction. There's a scoring function that tells them what the score is. So they get a higher score the more reactions that they study. So Rob did an experiment where so the video from I think that's some. So Robin did an experiment where he let master students go into virtual reality, I think 20 students for a half hour each. So 600 minutes in total, which is 10 hours. And then he looked at how he compared how standard automated reaction discovery algorithms compared to what students would do. And what he found was this graph. So this shows all the points that these sort of orange points are all the points that the students sampled during the 600 minutes. And then the green is the points that the automated algorithms. And so what you see is that the students are sampling everything that the automated algorithms are sampling, but they're also covering a much larger region of the configuration space. And so again, this is kind of what we expect, is that humans aren't restricted by the algorithmic rules that most algorithms follow. And so they can actually cover much larger regions of the configuration space in a shorter time than the automated algorithms. And so if you look at the paper, he actually shows that the humans pretty much discovered every single reaction that the machines discovered and more, which was a nice result. Here's another example where we're using iMD-VR to accelerate the binding of proteins with small molecules. And so this is a protein called neuraminidase, which is isolated. I think this was from the 2013 Asian flu outbreak. And this is a drug called Tamiflu, which was being used to target this molecule here. We were trying to understand how does Tamiflu bind to neuraminidase. And so this shows Becca docking the molecule to the protein. And you see there's a little flap that has to close in order to kind of lock the drug into place. So what she does is she opens the flap, and she'll be able to pull the drug out, and then she'll put the drug back in, and she'll close the flap back up again. Yeah, this is another example. This is kind of a molecule, a protein called trypsin, and a binder called benzamidine. And I say this is kind of the hello world of protein ligand binding. This is the system that whenever you have a new method for studying protein ligand binding, you always start with trypsin benzamidine because it's very well understood. And so we did the same thing. We looked at trypsin and benzamidine. So this is work by Helen Beeks to study the system and gives you a sense. This is using augmented reality overlay. So all the VR headsets now they offer augmented or virtual reality. So our app lets you do either one. I don't really distinguish between augmented reality and virtual reality because now all the headsets they do augmented easily. It's very easy to turn that on. So including the framework that we've developed that lets you do augmented reality. And so here's Helen trying to bind the benzamidine into the active site of the protein. You can kind of watch as she guides the molecule. There you go. So one question we have, and this is the last part of the talk. This is the last thing I'll talk about. And Harry will talk more about this. One of the questions is, so in a research context, if I'm able to, this is my trypsin protein and this is my benzamidine. If I'm able to create a path through this 6-dimensional space, so 3 translations and 3 rotations, right, that's what I'm doing. When I move a molecule out of the protein, I'm making a path in 6-dimensional space. And then what I want to know how to do is, well, how do I get some quantitative energy along that path? And usually what you're interested in molecular simulation is the free energy along that pathway, because that tells you about the kinetics, tells you how quickly things bind and unbind. And so you want the path in the 6-dimensional space. So this paper here, which we published a couple of years ago, was kind of the first attempt to say, okay, well, how do I take this human bias simulation and then unbias it to get free energy? And the method that we outlined was you basically take the trajectory in the hyperdimensional 3n dimensional space, and then you project it into a 6-dimensional space, and then you do a fancy kind of umbrella sampling along that path, and that gives you the equivalent. And so here's some results from the paper. So Helen, the lead author of this paper, she took the trypsin benzamidine and she generated 7 pathways to pull the to bind the trypsin to the benzamidine. And so these show you the 7 pathways. This is each of the 7 pathways that she generated. And then what she did was she was then able to calculate using 2 different forms of umbrella sampling. She was able to calculate the energies along those pathways. These are the free energies for each of the pathways. So like the green one is the green surface, the blue one is the blue surface. So each of the colors here corresponds to the colors here. And so the really cool thing was that when we looked at this in detail, the first thing is that the free energies that we got were basically what we would expect from a typical way of doing this sampling, right? Using standard sampling methods. So it showed that this way of doing unbiasing with iMD-VR works. And then the other thing we found was that if you actually looked in detail at the minima along the free energy path, you saw metastable structures that corresponded to what other people that have studied the system have seen, which was really cool. And we didn't know that in advance, right? So we kind of, after we saw this minimum, we looked at the literature and saw that for other structures, which was good. So that gave us some confidence that this could be a cool method to sample energies in other systems, and not some of the stuff that Perry and Ludovic are working on right now. So conclusion, I just want to say that like new technologies like VR and cloud computing let us interact with living and breathing molecular structures, which are animated using rigorous physics. And that these technologies offer some interesting new ways of exploring complex molecular systems. And yeah, we have this ERC grant at the moment called NanoVid, where we're looking at how to do this, combining kind of fast molecular simulation and fast analysis methods with GPU accelerated computing and real-time graphics and human computer interaction. So everyone from the group here, Mohammed, Luis, and Raz, Hugo, and Harry are part of this grant that we're working on right now. And we're going to hear a few more talks today and some demos tomorrow. Harry's going to talk about how to extract quantitative data from iMD-VR simulations. Ludovica is going to talk about applications to learn about binding energies in small ligand receptor systems. Mohammed will talk about using data from iMD-VR to train AIs. And then Raz will do a demo of understanding kind of what humans are able to feel while they're using iMD-VR. So yeah, that's all. If anybody has any questions, I'm happy to take some questions. Yeah, good. Maybe I don't have time. I can just probably talk.
No, we have time. Okay. So I have a question on my first. Also, we got some people online. They ask. So I can start with one question. So I didn't understand when you studied this binding of drugs to proteins. So should you know the active site a priori, and then you put the molecule in the right place, or so is there a way to, let's say, to do a kind of iterative docking or something like that? Just if you don't know the active site of
yeah. So if you if you don't know the active site, then well, it's just like just imagine you're not using VR and you don't know the active site. That's really hard. So it's still hard even with VR, but now you have more information. But if you're doing a protein ligand binding site study and you know the active site without VR, then you know then you have more information, right? So so we've looked at most of what we've done is we've done binding in cases where we already know or have some idea of what the active site is. But in principle, it would be possible to use this to try and find what the active site is. Because you could look at the structure and you could look at the molecule and you could say, okay, well, maybe it fits there, or maybe it fits there, or maybe it fits there, and you could try that. So there's no reason why you could not do that. But just like in the case where you do not have VR, it's just, there's a lot of possibilities.
And when you pull the molecule from the active site in this umbrella sampling, so how does it work? Because umbrella sampling, you have these windows and then you run for some time the simulation in each window. And here, don't do that, right?
Yeah, it's the same idea. Yeah, it's just a fancy form of umbrella sampling that was developed by this guy Kirill Zinoviev, but it's basically that idea. It's effectively that idea. So well, all you do is use the key idea here is that instead of starting from some general path, you start from a path that you generate in iMD-VR. And I think Harry will talk a little bit about this in his talk, but when you start with a iMD-VR pathway, you get is everything okay back there?
I think we are preparing to go to break.
We're about to take off. So the important idea is that when I generate a path in iMD-VR, there's already physics in the pathway, right? And I'm using my brain has a certain amount of spatial intuition that I'm using to generate the pathway. And so the path quality is pretty good, right? Because it already has physics built into it, because the physics is being simulated in real time while I do the motion. And so, if I start from one of these pathways, I can get a wide range of different pathways. You see that they have different energies and different microstructures. And then I can get, free energies along them. And these pathways are sort of like, they're physically informed because they already have the physics in them because of how the equations of motion are being integrated.
Yes, but if you pull, let's say, too hard, there is no risk to go through other, no minimum energy.
Yeah, that's right. There's a balance between, you don't want to put the force to like 1,000,000 newtons because otherwise that's crazy. So you want to be on a scale of the force, that's reasonable for the system. So typically the forces we're working are around tens of piconewtons, something like that, right? Yeah. So there's a balance. And so if you look at this video, if you look at this video, what you'll see as this video goes along is you'll see that actually one thing Helen will do is she's modulating the strength of the force that she's applying to the system, right? So you'll see that she pulls up a little, maybe she does it here. Can't remember where she does it, but she'll actually change the force. You can change the force on the fly using the controller. And that lets you kind of, change the strength of your interaction. So she's not doing it here. If she does it in a little while.
Okay, so you can scale the external force.
You can scale the external force, that's right. Yeah. It's something you can control. And when you do a study, it's something we'll have to look at. It's quantifying what the kind of force go on these pathways. Yeah. Here, I think she'll scale the force right here. Yeah, there. You see, she's changing the force. That's what she's doing. She's changing the scale of force that she's applying, so she can make it. If it's far away, maybe you can have a stronger force, but if you get very close, you do very small things and maybe have a weaker force.
Example to show that the students on the reactive space and passing the molecule that they found a much wider space than it.
Yeah.
But we are used to use the nanoreactor
Yeah.
to explore things automatically. I was surprised because I was expecting the students to be more biased by the chemical, not exploring the stupid possible reaction.
Yeah.
So it seems to be the opposite.
Well, I mean, they definitely explore some stupid reactions, but they also get all the reactions that the normal algorithms find, right? So they get, like, that's the whole point here, is they get everything that the normal algorithms find and more. So like, like, and that's, what's really cool, but the thing is also like, this was with, I think, 20 students, 30 minutes each student. And so that's like 10 hours. But when you run your Terracam or you run your automated search, your GRM or whatever, you typically run it for like weeks and weeks and weeks, right?
Use hundreds of
Using hundreds of trajectories in parallel on a really big computer. And so, what was crazy here is like 20 master students, half hour each, basically got the same answers as like my colleagues at Argonne National Laboratory who use huge supercomputers with DRM for months and months, and we got the same thing.
What we have did.
Yeah.
Use you to explore the Pacific.
Yeah.
Or maybe it's a white strategy.
We need master students then. We need master students that like VR, but I think, that's the trade-off. But yeah, like that was what was really cool about this was exactly that conclusion.
Which kind of computers we use for the
Oh, just like laptops with GPUs.
for this trajectory, trajectory for whatever.
Well, so the way that this works, maybe I don't have an image, is typically you have the headset. Well, you'll see in the demo, if you come to the demo tomorrow, you'll see how it works. But it's basically the headset is basically a mobile phone. All the headsets are just mobile phones. And then they connect wirelessly to a server that's running physics. And the server typically has like one or two.
Not very small compared to.
Yeah, compared to the computers that you're using, much. Yeah. Yeah, obviously, if you want to then do analysis on the results that are generated, so if you wanted to like, refine, maybe you want to. There's an important point here, right? What the students can do in real time, you're limited by what physics you can run in real time. So, you can maybe run like a low level DMT. So if you need to run some crazy coupled cluster thing, then obviously you can't do that in real time. So, if you would take what you probably want to do with the results the students generated, you probably then want to refine that at a higher level of theory. And then you still do that on your big computer, right? But the point is that the initial discovery phase, much faster, gets you pretty much everything.
I think it's what you're saying.
Yeah.
So when you run these demos in, let's say 1 hour, so for how long the simulations run? 10 or 12 seconds. If you do a classical simulation.
In terms of seconds.
Seconds.
Yeah, that's of course how much it's like that. Would say that if you are 30 seconds to 1 minute, it would be 50 seconds.
Yeah, 1 minute of real time is how many people said probably.
One second. About 3 seconds.
Okay,
So,
so 3 seconds.
yes, all the
One
over people said sense, but
Okay, so yeah, all right, so
This is a classical level, right? The horse paper.
yes. Yeah, but then the same is supposed to apply to the same. Like binding.
yeah. Pretty much, the time set's pretty much the same, whatever simulation. I mean, your time step is limited by the time scale of a molecular vibration.
Yes.
And for it to be a fluid experience, it has to basically run at 60 frames per second. So whether it's a quantum simulation or a classical molecular mechanics simulation, your time steps usually on the order of 1 or 2 times a second. And you said 1 second is
Yeah, with a 2.7 time step, and with the things that we use, like sending frame every 5 simulation steps instead of sending it every simulation step, you're looking at, yeah, on the order of 100 femtoseconds per second.
yeah.
We did several nanoseconds, so we have to do full day.
I think you don't want to be in VR a full day.
Yeah, sure.
Like that's not a good idea.
Yeah.
So you'll get uncomfortable. So, the thing is that you can get over very high barriers faster though, because you bias in the direction that you care about. So
Mhm.
like if I was to like look at the barriers here are like look, that barrier is like almost 60 kcals per, like some of these barriers are like 30 kcals per mole,
Mhm.
right? So if I was to run a if I was to let that reaction happen spontaneously, I'd be waiting for years. But the whole point is that in iMD-VR, I can change the force, and I can just do it. So it goes faster, right? So I don't need to be, so you have to, the time you're in, you can make things happen faster in iMD-VR than they would happen otherwise. That's the whole point, right? So you don't want to be in VR for a whole day.
Yeah.
You won't feel good after.
I think the problem is I still think it's an equilibrium simulation, but
Yeah.
it's not an equilibrium.
It's not an equilibrium. That's the whole thing. Yeah, exactly.
Okay, perfect.
And if you want to run very well solution, run it in touch with the snapshot state of the system, and then continue from that particular instance and whatever there is. Because you don't want to spend 3 hours.
Oh, sure. Yes.
With snapshot say that
Okay, so thank you, David. It was a very nice time. Now, so now Mohammed, what stage is do it? So we will talk about exploring strategies for machine learning in interactive molecular dynamics virtual reality. So if you want, need 2 minutes for going to the bathroom.
Yeah.
Here on the left. So we can we wait a few minutes. Yeah. So you know how to.
sure.
And then let's see the. And then let me replicate. What is it? What? Yeah, we need to replicate it because otherwise.
Oh, yeah.
Here in the configuration.
Duplicado?
Yeah.
Oh, no. Sí. To exit and do it again, right? No.
It's it. That's great. I'm going to go on the back. So we developed the machine learning part. No? So I was checking if there's also outside ones.
So my talk is gonna be about exploring strategies for machine learning and VR. First of all, I want to thank you for inviting us here, and I thank the audience for being present and assisting to this presentation. So what is NanoVera iMD-VR, or what is the software that we are developing? As we've seen from Dave's presentation previously, we have developed the software is called NanoVera, and that is an interactive molecular dynamics software that lets people do interactions with molecular dynamic systems in virtual reality. So it's an open source software. Publication is undergoing, but it's available on GitHub. NanoVer combines high performance computing or the use of GPUs with VR interfaces to enable real-time interactions with molecular simulations. What are the advantages of doing iMD-VR? We as humans, the claim is that we have some sort of spatial recognition system, and we live in 3D world, so we do really good at interacting with 3D objects. NanoVer uses real-time physics simulation. So as Dave mentioned, we are not using game physics, we are using an actual molecular dynamic simulation engine. In our case, we have support for two engines, Superman and AC. It lets people be collaborative and have a multi-user experience in VR. So two or more people can see the same system in VR and interact with it at the same time using different VR bitsets. It is really good at accelerating the event sampling. As Dave mentioned, well, we want to bias the simulation towards a particular reaction system or a particular sampling condition. We can do that using NanoVer. This is the basic architecture of NanoVer. So what we have here is we have a server and we have the VR client. So the server here is plugged into a molecular simulation engine. For our case, it's either OpenMM or AC, but the software is consort, so you can plug in whichever molecular system that you want. iMD forces are being exchanged between this server and the molecular simulation engine. And on the other side, with the Python client, if you don't want to do VR, you want to use Jupyter Notebook to do the manipulations or interactions or whatever processing you are casually doing or normally doing, you can do it on a Python client. Or if you're doing this in VR, there is a VR client. So the VR client uses iMD interactions, which get transferred via the NanoVer server as iMD forces into the molecular simulation engines. Calculations happens, and then we have the molecular simulation data that is fed into the VR visualization or the VR interface. So why am I explaining this? Basically, NanoVer is a tool that lets you either collect data, generate, and collect data. So you can generate whichever systems, if you are capable of putting it within the limits of the computational capacity that you have in NanoVer, you can simulate that system, interact with it in VR, record the data using the server, and have it for post-processing whenever you want. So what are the current challenges to this? First of all, there is a computational and temporal cost because simulations are limited to relatively small time scale due to computational constraints and due to people not spending too much time in VR. You can't spend 10 hours in VR collecting data. So there is a computational and temporal cost. It requires specialized knowledge to effectively manipulate molecular systems, but we'll go back on that. So as one question was earlier, for a protein system to bind a drug into it, you need to know where the binding site is. And that requires special knowledge. But there are systems where humans innately know what to do. Like if you show them a molecular system and you don't say anything, and I've done this with a couple of simulations, they just go in there and do the thing without any instructions. And I'll show an example of that. And then you have sampling, so it's very difficult to access rare conformational states and transition pathways in regular MDs. So if you want to sample rare events using classical MD approaches, as we spoke about earlier, it would take a lot of time to generate those pathways and to identify which ones are rare and study those. So here, iMD-VR would be really beneficial. And extracting meaningful insights from high-dimensional trajectory data. So, as I said, we can report all the trajectories that we generate in VR and then collect those and do post-processing on them. So, here the question is, what is the potential of AI in this system? Since this system is generating and recording data, we should ask what data do we have there and how can we integrate it into an AI approach. So these are three systems that I'm working on two of those currently, but I'm hoping to be working on the third one soon. So the first one is a carbon nanotube and the methane molecule and the sort of molecular channels. We're trying to simulate molecular channels. And the goal of this task here is to thread the methane molecule through the nanotube. And this is one of the examples where humans do it really good. Like, you don't even have to give them instructions. We ran some demos in the lab, and they just put people in the air and tell them, what do you want to do? And they just do that and pass it through the nanotube. The second one is taking 17 alanine peptide and then trying to tie a knot within it. Why is this important? I'll be talking about that in the second part of the presentation, but it's very common to have knots in proteins. And the third system is a slightly more complex system, and that's docking a drug into a protein. So why is this more complex? Simply because this would be just a simple path. Like if you can align this molecule with the entrance, that's it. The problem is solved. You just drag it that way. This is slightly more complicated because you have to form a knot, and there isn't one path to form a knot. There is a multitude of paths, and we want to explore those paths and the ways knots are formed in the protein. And this is a bit more complicated because different paths to get into the binding site generate different energy profiles, and the way humans do it might not be the best way or not. So the first part I'm exploring in my research is to do imitation learning from expert recorded sessions in iMD-VR. This is basically learning molecular manipulations by observing experts in iMD-VR demonstrations. So I put people in VR, ask them to do particular tasks, record that data, and then use it to train our agents to try to do the same as humans do in VR. The concept, as I said, is to train AI agents to mimic the expert manipulations. The data sources are data generated from the NanoVerb framework. The key techniques here, there is behavioral cloning, inverse reinforcement learning, generative adversarial imitation learning. So the difference between the three of these is behavior cloning is just telling the system to copy whatever it sees. So say it sees 10 trajectories, and then it tries to copy those 10 trajectories. But if the data varies too much, or if the initial start varies too much from what it saw in the data, it will not solve the problem because it's only doing cloning. Inverse reinforcement learning is a method developed to try to come up with an explanation or a policy of how that task is solved. What is threading pollution through an nanotube? What is tying the knot? So we will have an equation that will map out the progress of the task. So you're halfway there, you're complete, you're at the start, you're way too far, you're way too close. So it's a mapping, sort of a mapping function of the task that you are doing. Generative adversarial limitation learning is generally used in gaming setups where you have an agent playing a game, and then it either plays against itself or plays against another agent. And you want them to learn from each other's moves. So it's no longer using humans, it's using two agents to compete against one another to have better results on the sampling. So what are the advantages of this? There is no need for explicit reward system because when we deal with reinforcement systems, usually we need to write down the reward function that lets the system learn. But using these, there is no explicit reward design. It leverages human expertise, and this is by collecting data that humans generate in VR. And the claim here is that it's generalizable across multiple systems. So the recording of sessions in VR between people and agents, or just recording data from humans. So none of it captures force applications, trajectory changes, interaction patterns, and decision points. So what we have here are mainly three types of data. So we have spatial features, which are the atomic positions, the distances, the angles, any spatial data that we can collect of the system. And then we have temporal features like velocities, accelerations, interaction sequences. And then we have chemical features like what are the elements in the simulation, bonds, electrostatic properties, energy profiles, potentials, all that. The challenges here are data scarcity from limited expert sessions. So the biggest issue with machine learning is, do I have enough data to train a model? How do I get enough data to train a model? So the challenge is, how can we get enough data from experts or non-experts to train our systems? Ensuring data quality and consistency between the data collection sessions, handling high-dimensional molecular representations. So how do we deal with that using imitation learning? So AI agents are capable of autonomous molecular manipulation without human intervention if we succeed into training agents that are capable of doing these tasks. Data generation is no longer an issue. We don't have to put humans in VR and ask them to solve the task. We can put some agents in parallel on a GPU system or in a laptop or a computer and let them run all the sampling that we want. It's a faster exploration of conformational spaces and binding pathways. This is what I was talking about. Human AI collaborative systems for solving complex molecular problems, since Nanover is a multi-user capable software, so we can put humans, one or more, in VR and have them manipulate the same system. We can also put humans with agents and get them to help each other manipulating the system. So, as an educational tool, it can be used for training systems for new researchers to learn molecular manipulations. That if we have people who are initiating the experience with how to do molecular simulations in VR or how to manipulate molecules in VR, we can have agents as tutors or as educators that show people how you manipulate the system in VR. So preliminary results from this approach have been published in a workshop in last year's API conference, and an extended paper is about to be published in the next few weeks in Springer Nature Computer Science Journal. So this was preliminary results on the behavioral cloning approach. What we did is we used a convolutional neural network to get the particle positions and try to predict the interactions, position, and scale that users did in VR. The results are very promising. The system was able to learn and map how to do the magnitude test. However, as I said, if it's too far from the training data set, it was unable to do was unable to solve the task if the initial conditions were too far from the training data set. What I'm working on right now is, since I said the issue with machine learning is to collect data, what I want to do is to create this nanover ML agent package that will be a package associated with nanover that lets you use PyTorch and Gymnasium as a. This is an environment creation tool for reinforcement learning and other AI algorithms, and it would let you create gym environments or gymnasium environments to train your models, process recorded demos, and train IL models. In order to do so, what I started with is this. So now I have a Python recording client that connects into Nanover while people in real time, while people are doing molecular simulations, reads the data from whatever they're doing in VR through the server, and through a task logic monitor, try to map or facilitate the task process. So, for example, for the nanotube test, I will show a video of this. So for the nanotube test, it will show you, this is a recording of someone doing the test. So it will show them that this state is start, now they're aligned, now they're inside, now they're exit. So it will change the visuals the better you're solving the test. So if you're at the starting position, it will be certain representation, and then if you get aligned with that, it will trigger another visual. If you're inside another visual, and then if you're outside another visual. And I keep track of all those states for the training process. So you're outside, you're aligned, now you're inside, and you're outside, and I keep track of all those interactions. For the knot tying example, I'm using the tool to in real time detect when a knot is present in the 17 alanine peptide, and then give you a couple of seconds to stabilize the knot and then stop the simulation. So these two tests are the demos that I will be running tomorrow to collect data from participants if you want to participate. So yeah. With that system, with particularly the knot tying system, it is a bit difficult to detect knots in protein peptides because, mathematically speaking, a knot can only happen in a closed system. If the system is open, that's not a knot. Whatever you form in it, that's not considered a knot. So the system has to be closed on itself. There are multiple ways to detect knots in systems in general, or in topological systems in general. There is these types of knots, so the system has to be closed, and that's considered an unknotted state. This is the simplest approach, which is the trifold knot, the one that is very common, and it's considered the 3-1 knot, and there are multiple variations. So protein knots are topological features where the protein chain threads through a loop, because it's not a closed system, it's an open system. So if it threads on itself, that's considered a knot, just like this, forming a mathematical knot. But we need to calculate a connection between both open ends of the system. So the biological significance of this is that it's enhanced protein stability against thermal fluctuations, or at least some literature claims this. Then functional implications in enzyme activity is another biological significance of knots in protein structures. The detection challenges here arise when we have a computational complexity of topological analysis. The bigger the system is, the harder it is to detect knots in it. There is an ambiguity in knot definition and boundaries, since, as I said, mathematically, a knot can only happen in a closed system, and we're dealing with open systems. So how do we close those ends to get the knot to be recognized? And there is a real-time processing requirement. So the current methods are mainly the Alexander polynomials. And while very accurate and very precise when they detect knot, the issue is that they have a high computational cost. So they're very expensive computationally. What I propose we do is that we use neural networks and precisely transformer architectures, and these are the backbones of ChatGPT and large language models. They were based on transformer architecture. And I want to use transformer architecture to feed the protein structure in and output what type of nut is present in the system. So for example, this is, well, what do you think? Is this a knotted system or a non-knotted system? This is the crystal structure of human methionine adenosine transferase. Well, this is a knotted system, and it is 3-1 knot, a trifold knot. So if I start simplifying the system, it is very clear that it's a knotted system. That's how it's done. My project in this is to create, as I said, a transformer architecture that would take in the coordinates and the bonds present in any peptide or topological protein structure and try to classify what type of knots are present in that system. So adapting transformer models for real-time protein knot detection, self-attention mechanism. So this is the thing that transformers are known for, that they have a self-attention mechanism that captures features that are way back. They have a better long short-term memory than another knot. I use feed-forward networks for feature processing. So this is basically the transformer architecture, and this is a feed-forward network that will get these latent features and transform them into a classification process. So this is basically a classifier that will let you know what type of knots are present in the system. I'm going to use layer normalization, but this is getting too technical on the architecture is to use this layer normalization to get coordinates and bonds. This is an approach just to optimize the calculation because you have the coordinates in values that are represented in a big representation. When you do multiplication and mathematical processes on them, they tend to get way too big, and that makes it difficult to calculate them. So normalization is the process of just getting those coordinates and those bonds and normalize them before passing them to the transformer encoder architecture. So this is what I'm working on. So these two systems, the protein knot detection system and the imitation learning process, I will be running a demo tomorrow to collect data for both these systems. So each session is a 30-minute session split into the first half is feeding the knot through the nanotube, and the second half is doing the knot time test in VR. If you want to participate, this is a QR code to book a time slot for tomorrow to participate, and that's basically what I'm doing.
And for the demos, maybe we can talk tomorrow. We have a list of people that is interested. We have around 18 people interested, so we can organize it later. The time slot. So, question? So, one thing, the machine learning thing. So you also use see the data from the virtual reality simulations for feeding also potentials, right? For developing potential or not?
I'm not doing that, but it has been done, yeah.
Okay. Here, so you need, as you said, a lot of data, right? But these virtual reality tools, I mean, probably you can run, as we said before, shorter simulations, right? Because so it's possible, let's say, to develop a machine learning force field entirely with this with this technique with the reality data.
It is doable, yeah. But the purpose of doing this is that we want to create a system that is in parallel working with humans. So if we use humans to collect data or to generate data in VR, and we want to have way more data than what they do, we can use that tiny sample of data that humans generated to train models that will create a very big data set that we can use to fix losses. So the ultimate goal is to get agents to generate data for us instead of humans.
Okay. So tomorrow you said you were going to do two things. So one is for the dots, right?
Yeah, this system, the nanotube, through the methane through a nanotube system, and not tying the 17 aluminum.
Okay, good. So that's one. Okay, so maybe now we have here outside a coffee break, and then you said people more will talk, right?
Yeah.
Okay, two.
Highly improvable, yeah.
Okay, good. So we stop for half an hour, have a coffee, then we come back. Then we talk about the organization of the house for tomorrow.
Okay.
You don't have the video. The machine show. If you don't put the field of it, that's like, well, I think for like, so it's just like inside the world, you have that field of the central part of the farm. Like, here, personally, make it real cool. Here's questions. Okay, when you go here, which takes really highlight that, like, in fact, your talk is just split. Your talk is effectively explaining that, you know, like that's what the talk is about. So the whole intro is explaining that we have talking about. So no, it's good, it's very nice, it's nice, but I'm big for it. So, it's all stuff I see that in terms of ways. Like, I see maybe, that something happened, but with no person. So, oh. You describe the little server organization. I didn't put that, so was I sure still with one guy.
I would like to use
No, it's really good. I think I told this before. I don't think we're about class one. I hope you're not used to. So. I guess good. I mean, all we really worrying about is 3 to 1. I would. What class would not? It would not. A lot of things, what class would not for this? I want to look at synopsis. I'm not to be on. Or we want to find for that. I got to be like this. I got to be like the neighbor. We do both things. So, I'm spaces and kind of still. About right, that's exactly what I thought. I mean, I really started. Guys, I don't, I feel like not trying to build my top box. Using these things, but. So. I put my followers for a free not. So yes, we want classifier, but we don't need something for everything else. We just need to be one, because if we spend a bunch of time doing for all of that, it's gonna take a long time. So like, in fact, is that in nature. Three one is like 99% of all the knots, so you spend a whole bunch of time to do that for higher order knots, they still exist naturally, because they don't exist for basic permanent. Required to start with. It's just so much better you ever got, yeah. So that's why my nature does make it close. So that's just like, I don't like this one here, but I'm trying to do that, that's a stick right. It's like, it's cool, but it's more of a mathematical problem. Instead of a. Like.
This is the. That's one. This is the one.
I like the video. Cool.
Using alpha. Try to pull on the parts of the system.
Very cool. So, you know, that's a great video. So so you're talking a lot better. It's like the size of nice, it's clear. But the thing is, you need to have. That needs to be like, effectively your talk is explained that. That's what people also, it's based on the value, potential paper, however we make it. Or. Yeah, exactly. They feel like obvious, like person water. Maybe nobody doing that one, or you're quite good at fingers, you know, each. This slow, this. So I'm flat. Yes, that might be the response. We talked about data classes, we didn't talk about force or. Yeah, I mean, I know. Yeah. Nobody here probably noticed it, but. I put some time back from. Those are big. Yes, I was trying to do first of these. I thought your categorization was good, so it's that, so. So, okay, so I just want to make sure you didn't forget about. They're all functional. Okay. So I just want to make sure you don't forget about. I think it's smaller than this, but. I think it's. Yeah.
Una reserva aquí vamos a acabar absoluta para frente, solo quedan dos charlas porque no digo que si quieres quedarte a escuchar un solo de esto. All these students are here. All of this students. Students still first and second. Oh, there's. You are for people on the same time you were.
So what is the structure of the tweet?
Here, the moment, I'm so thinking of it. That's so reality. Do you teach class? Sometimes we got. Computer vision. E vai, segue a consideração de Taer, essa foi a barriga in fact, the software that we use to write sensing the open source framework called Open Frameworks, C++. Does a lot of things, a lot of physical effort. So it was just one thing for students to even run like a small workshop to teach them how to sometimes many of these students have some and open framers, very cool. This is foundational. So all of the things, all about science here. That's all here. Before you're. Exploration of actually university. Developing these. Where the students working on the cancer, on physics. Using data. They love. Standard fun, they already say that. Then many of them. That result is. Look nice, right? That feel. Those types of some of the students, we also felt that. The temperature a little bit in here. It's a little bit.
It's cold or is
It's a bit too hot.
okay?
Is that just a little bit? Is that all right?
Yeah, sure, sure.
Like maybe because we're used to lethal.
And we are 26 degrees here. No, now we're decreasing.
Yeah, we're melting a little bit. We're trying to ask for a little bit down there. It was 26 degrees.
Most of our students are online apparently, but they will come tomorrow for the presentation, but they are now listening to the talks by YouTube.
Cool.
Okay, so let's continue. So now we have two talks, right? 100 plus. So
Ludwig.
Ludwig, sorry. So now let's go with Harry's talk. So Harry Strout is a postdoctoral researcher, right, with David in Santiago. So he obtained the bachelor degree at the University of Bristol, right, in chemistry,
Yes.
and then the master studies in theoretical and computational chemistry at the University of Oxford. Then you come back to Bristol for doing the PhD, right? Also in computational chemistry.
Exactly.
Okay, perfect. And now, so how long do you work with David? So a couple of
I've
years.
been in the lab a year since I arrived April last year, so just over a year.
Okay, great.
Yeah.
Okay, so we are looking forward to your talk. Thank you, Harry.
Thank you very much. Is it Miss Elizabeth? Or is it the other one?
Okay, so
Cool. Fantastic. Well, thank you very much. Well, so thank you for coming. So yeah, today I'm going to be talking about something I've been working on recently to try and take iMD-VR and use it to extract quantitative information about systems at equilibrium, or how they would behave at equilibrium, but from non-equilibrium simulation. So I'm going to talk about why we want to do this, go through the developments of the method that I've been working on, and then give the preliminary results of the method and see how it goes. Cool. So in our group, we have this tool, interactive molecular dynamics, that or iMD-VR, that Dave introduced and Mohammed talked about as well. So I won't go over it in much detail, but essentially it allows us to interact as researchers in real time with a molecular simulation. And it's very easy to make the system perform rare events, undergo rare events that wouldn't usually happen for much longer time if you were to run normal simulations under equilibrium conditions. So it's been used in the past to do drug docking, protein folding, and chemical reactions. I think at least two of those they talked about. And it enables you as a researcher to apply your chemical intuition to understand systems and manipulate them. The software that we have to perform iMD-VR is Nanovel, which Mohammed explained. It allows us to form multi-user real-time interactive dynamic simulations in virtual reality. It also delivers quantitative information about the simulations that we're performing. So stuff to do with the molecular system, like positions, forces, velocities, energies, etc. And then also stuff to do with what you're doing as a user to the system. So the perturbations that you're introducing, the forces that you're introducing, the relative potential energies, and the work done on the system that you're inducing via the manipulations that you do. It interfaces with lots of other common softwares, including OpenSE, OpenMM, ASE, MD-Analysis, and MDLB. So this is an awesome thing and an awesome tool that we can use. But the problem that we have is that iMD-VR is a non-equilibrium enhanced sampling technique. So we're driving the systems that we're interested in far from equilibrium in the manipulations that we're doing, introducing strong perturbation system that mean that we make processes happen on much shorter time scale. So a process like protein folding could take something on the order of milliseconds to achieve, whereas we're forcing it to happen on time scales to be a repeat second. So like 10 to the 9 times faster. To give you an idea of how much faster that is, that's like doing something in 1 second that would usually take about 32 to do. So it's way, way quicker. And that means that we're far from equilibrium, so far from the way that the system would behave if you were to leave it and simulate it in its natural environment. And this makes it difficult to recover information about how the system would naturally behave without the external perturbation, which is what typically we're really interested in. We want to know what the energy barriers are for particular reaction processes and be able to calculate the free energies and rates for processes and things like that. So the question then is, how can we use this software, iMD-VR, to be able to quantitatively describe processes in molecular systems at equilibrium through non-equilibrium simulation? The toolkit that we would like to turn to be able to use the toolkit to employ in these situations is non-equilibrium statistical mechanics. In particular, the Jarzynski equality, which relates the work done on a molecular system to the difference in free energy between states of interest. So, yeah, I've got it here. It relates the irreversible work being done by an external force to drive a system from an initial state A to a final state B in a finite time. And it relates that to the difference in free energy between the two states. This, on the right-hand side of the equation, there's an overbar, and that denotes that there's an averaging going on here over an ensemble of trajectories. So you do lots of different trajectories, I think formally an infinite number, and then from that ensemble average, you can determine the free energy difference between the states. And it's not dependent on how quickly you do this process, which is good news for us, because obviously we're doing things very quickly. So can this be applied directly to iMD-VR? Sort of no. And the reason is not that we're necessarily doing something unphysical or something wrong in our simulations, although we're obviously adding a lot of force in. The problem is that we need to average over lots of trajectories. And to do that, you need to keep the reaction coordinate that you're interested in constant and the time that you're doing constant. And it's very, very hard as a human to replicate the same movement twice, let alone approaching an infinite number of times, without deviating in either time, space, or example. So, is there another approach we can turn to that would help us here? And the answer is yes. Steered molecular dynamics, it's like the cousin of interactive molecular dynamics, except in this case, you pre-program the reaction coordinate that you want the system to follow, and it's essentially the time that you want it to follow it to. So it's still a non-equilibrium enhanced sampling technique. You still apply external forces to the system. You still produce trajectories that are out of equilibrium, but now it can be used to recover the equilibrium properties, and this has been shown by people in the past. But the problem with it is that we now need to define in advance what we want our system to do. And that may be a non-trivial thing to do if you've got a very complex reaction coordinate and you're not exactly sure which motions affect the dynamics of that particular behavior that you care about. Very hard to program that a priori. So, is there a way that we can get around the limitations of these two methods to combine their strengths, combine the path sampling ability of the SMD method with the ease of defining a reaction coordinate using iMD-VR to produce a method that can ultimately recover equilibrium properties from non-equilibrium state? So that's what we're going to try to produce. Well, that's what I'm trying to produce. To assess the performance of this method, there are essentially like 3 levels that we want to try and achieve. So the first level is like, can we get qualitative results? So the things that we produce have the right shape, they give approximately the right differences between things. The second level is, can we obtain quantitative information about it? So if we have a reaction coordinate, can we quantitatively calculate the differences in free energies along that coordinate? And then the third level is, can we get information about the most likely from these things? So the minimum free energy path, can we quantify that? So now I'm going to talk about the method itself. So the ideal workflow is to go into VR, form an iMD-VR simulation to create a trajectory that defines the reaction coordinate that you're interested in. Then, secondly, extract that trajectory and define a reaction coordinate from it. And then use that reaction coordinate to form repeated SMD simulations, which you can then calculate these equilibrium properties from. Fortunately, recording the trajectory is already sorted for us. We can already do this in Laravel. So the next point to think about is, how do we extract that coordinate? And there are 2 potential choices that we could make about how we define the coordinate from an iMD-VR simulation. So one is to take the position of the user interaction itself. So as you pull a molecule, follow the position of your hand and use that position as the thing that's going to define where you're applying the force from during the SMD simulations. Or should we take the positions of the atoms that you interact with during the simulation? And the choice that I've made here is to use the position of the atoms. The reason for that is that the motions of those atoms that you're interacting with are still driven by the chemical intuition of the researcher, because you're still defining the reaction coordinate yourself. But there's physical information about how the system wants to undergo that process, encoded in the way that the atoms move during that. So if the researcher pulls a molecule to place in the system where it doesn't want to be, it won't go there naturally. So it will avoid regions that are very unlikely to be sampled naturally, which is good for us. But the raw trajectory will contain noise in the positions or in the coordinates, due to the fact that when you're simulating these things at finite temperature, your thermostat is going to be kicking the atoms about, which basically means that all of the paths that they follow will be jagged and not smooth. We want them to be smooth in order to perform SMD simulations and get quantitative results. So the solution there is to take the raw trajectory and smooth it, essentially. So interpolate those points and using a spline and interactively smooth this trajectory to the point that a user defines. So I've written a class to be able to do this, that basically allows you to load in your trajectory, which is the sort of slightly faint wiggly line you can see there, and then smooth the parameter. You use the parameter to smooth the curve arbitrarily and change the start and the end point, so there's noise at the start or the end of the trajectory, and change the number of points that you want to define that trajectory, which is important because the number of points that define the trajectory will define the speed at which your force is going to move along that trajectory. And the slower your force moves, the easier it should be to converge simulations. So having the ability to change that number of points. So that's that bit sorted. The final bit is to try and actually perform these SMD simulations. To draw from previous research in this area, we go back to Jarzynski, and he identified this equation here. This is an equivalent form which Jarzynski follows it, but it more clearly relates the free energy to the ensemble of work values. And this is a useful equation because if the work distribution that you're sampling from is Gaussian, then you can write this equation in terms of an expansion, which is curtailed at second order. All of the other terms evaluate to zero. And so you only need to look at the average of the work done and the variance of the work done distribution. So that's kind of cool. We'd like to be able to use this. Then in around 2003, 2004, Park and Schulten devised this approach that uses this expansion and applies it directly to SMD to calculate the free energy profiles of reaction coordinates that they're interested in. So they used a harmonic restraint with a stiff spring, and this ensures that your distribution is going to be Gaussian, and basically means that you can use this expansion shown on the previous slide to calculate the differences in free energies between different states, which is super cool. And they applied this method to a simple, effectively one-dimensional system of decaalanine, and they were interested in stretching it from its natural helix structure to an extended orbital structure. So pulling it along a 1D group. And they found that with as few as 10 steady molecular dynamic trajectories, they could get very good estimates of PMF, which is really exciting because that, in theory, doesn't take very long to do. So we can get quick and accurate results from an approach like this. So we'd like to be able to emulate this, but there are some things that we need to think about if we're going to apply it to an SMD approach that comes from iMD-VR rather than what they did. We will need to constrain part of our system, so fix it in space completely, while we pull the other part. And this is to essentially prevent the whole structure just being pulled along through the simulation, and means that you can actually sample the reaction coordinate because it creates a relative coordinate from the absolute positions of the simulation that you're performing. We also need to slightly tweak some of the equations they have so that we can apply what they use for a one-dimensional line or one-dimensional reaction coordinate to a 3D curve through space. And we need to make sure that the reaction coordinate is smooth for the approximations to hold. So this is a kind of sketch of what we're going to end up doing. We have this curve through space where the curve defines the position of the force that we're applying, or the position from which we apply the force to the system. At some given time, the atom that we're interested in applying the force to will have a position, and the force that we are applying is moving along this trajectory at some constant velocity. So we can calculate the force that we're applying, the speed at which this restraint is moving, and that gives us the work done over time. We can use that as our free energies. So here's the workflow. Load the system, apply a slipstream harmonic restraint, equilibrate the system with the restraint to generate the initial distribution that you're going to sample from, which should be Gaussian. Generate an ensemble of starting structures within that distribution, and then run simulations, S&D simulations for each structure, and calculate the work done for each of those. And then you can use that data to do your averaging and evaluate your properties. Okay. There we go. So now we've got the third step sorted, we can start to think about how to actually apply this. So this workflow is implemented in a development branch of Nanoware, and it will be made available so that we can use it open source. Cool. So how does this method actually fare? Let's take a look at the same system that Park and Shulton examined, this Decker-Allenian system. Again, we want to use a simple system that we can quickly evaluate and understand. And it's nice that they've already done it, so we have kind of an idea of the benchmarks and the types of techniques to employ. The reaction coordinate, again, is just this stretching from the coil structure that you can see here to essentially an elongated version of this. And it's a simple reaction coordinate in terms of the distance between the two ends is one-dimensional. You just increase that distance. So it only depends on one parameter. But it's complicated enough that you've got non-bonding interactions, and there's a structure that a natural structure that doesn't depend on the exact sort of topology of the system. So it acts as a nice proxy for something like a protein folding system, which are the kind of things that we'd like to be able to extend this method to. So the workflow here is going to be to constrain our system first. We're going to take nitrogen atoms at either end of the molecule as the things that we want to keep fixed and pull. So we constrain one of those by setting its mass to 0. That means that its position is not going to be updated by the integrations. We then go into iMD and grab the other nitrogen atom, pull it along the trajectory. You only need to do this once. Then from that trajectory, we're going to smooth the raw atom path. This is an exaggerated version. In practice, you wouldn't want to smooth it to get a straight line. More like you want to just eliminate some of the noise. And then we're going to generate the starting structures and run SMD on each of those structures. Cool. We're going to compare this with doing a collective variable-based approach. So rather than doing things in terms of atomic positions exactly, collective variable approaches take some internal coordinates of your system to define the types of pathways that you're interested in. In this case, we're interested in the end-to-end distance between the two nitrogen atoms. So that's the collective variable we're going to use. We're going to pull the system, quote unquote, pull the system along the same coordinate using that collective variable. at the same speed as we do for the iMD version. And we're also going to use this to calculate the ground truth that we're going to compare to, because the result that you should get with this system should be essentially the minimum free energy path that the system wants to take. And we're also going to compare a linear trajectory through space. So we've got this iMD path, which isn't going to be perfectly straight. We want to compare it with a linear trajectory, which is like a naive guess of what the minimum energy path might be if you thought about stretching a system from A to B. This is pre-programmed and provides a naive guess for the reaction coefficient. And it's nice because it also uses this same S&D approach we've developed for the curves to evaluate this version. And it relates to what path we should. So here are the results. The red line here is the ground truth. This is the work calculated from a reversible pulling simulation, which basically means that the work done in this case is the same as the potential of need force for the free energy profile. And then in green, you've got the collective variable version. In this light blue, you've got what we get from iMD. And in this dark blue, you've got what we get from the linear trajectory. So first thoughts are they all work quite well, or they all seem to work quite well. These are from very, very limited sampling sizes. The linear trajectory was performed 10 times. The iMD trajectory, 8 S&D pulls, and the collective variable one, again, 10 pulls. To take a closer look at, in particular, how the linear trajectory and the iMD trajectory compare, because the collective variable trajectory, as expected, is the same, essentially follows the same path throughout truth. So looking a bit more closely, we can see that in the linear case, there is a slight divergence at the start from the ground truth. There's also a slight divergence at this shoulder here, and then at larger distances, it again diverges slightly. And what's interesting is when you look at the one from iMD, it kind of fixes the stuff around the start and around the middle, and then only kind of diverges towards the end. I want to stress that this is from a very, very small number of simulations. So in order to properly analyze this, we'll need to run a lot more trajectories and generate the proper distributions. But this is super promising because all of these calculations from start to finish, I did on my laptop, including, so from putting on the VR headset and pulling a molecule to generating these plots, it was an hour, which was nice. So in our assessment criteria, we have 3 levels. Level 1, can we obtain qualitative information? Well, it's the right shape, so yes, that's cool. Can we obtain quantitative information? These PMFs are following pretty much the same curves, so we think yes. Can we obtain quantitative information about the free energy profile along the most likely path? There's a slight discrepancy between the iMD version and the ground truth. So we might need to do some kind of optimization procedure on the path that we do from iMD, but this is kind of to be expected. It would be weird if you pulled the thing along exactly the right path the first time you did it. So this seems like a win for this method, essentially. So the next steps are to thoroughly test the method, to extend it to more complicated systems. So in Ludovic is going to talk about this maybe, so I won't explain it now, but one system that we'd like to look at is the Glowacki receptor, which is like a receptor that selectively binds glucose more strongly than other sugars. And also, this last point, we'd like to investigate methods of optimizing these paths that we get out of iMD. The initial results seem to suggest that we get something that's better than a naive pre-programmed guess. So can we use that as a base to optimize to find the minimum free energy path and then do the same procedure along that to guarantee quantitative results about with barriers that are representative of how things really behave in the real world. With that, I'd like to thank everyone who's worked in the Glowacki group on Narrowbear and previous iterations of the software. I'd like to thank the funding bodies that have funded this research and Pitius and the University of Santiago de Compostela for hosting us. Then I'd like to thank you guys for having us here today, and thank you all for your research. So now
You have more questions?
I signed this, something, this is for 8, just one iMD-VR path,
so this is one iMD-VR
but
poll.
then sampled at 8 star geometries.
Yes,
Okay,
exactly.
sorry. So that took you an hour? You're saying an hour in VR?
An hour. No, an hour from when I did the poll that took 30 seconds.
Right, okay, to generate the initial path took 30 seconds.
So that's 30 seconds.
Right,
Pulling that out and smoothing it for minutes, running the SMD, which actually ran 10 times, but in two of the trajectories, the system buckled, which is what I saw in the collective variable version and the linear version as well. Hence only taking the 8 where it didn't buckle, and so where it effectively maintained its helical shape and stretched to forward smoothly rather than sort of starting to stretch, buckling up, because that's not really actually what we're interested in.
well, you would see the potential energy, I guess, shoot way out.
Yeah, exactly. And you can see it in the pre-image profile.
Yeah,
So it's easy to see what an anomalous result is in this one.
okay.
And it's not a fault of the method itself, I would say, because the other things, the other benchmarks also show this behavior. It's more a problem with the system that we're going from some non-equilibrium point to a non-equilibrium point, and at that start point, you've squashed the spring down into an uncomfortable place. So that's the sense of that. But if you're interested in going from an equilibrium point to an equilibrium point, like a binding socket to somewhere in your solvent, then you shouldn't have that problem.
You go back,
Yeah, sure. Half
go
that.
back. Yeah, those plots. Yeah, I didn't understand what linear trajectory SMD is.
Right. So that is basically the same as iMD-VR, but rather than taking the path from an iMD-VR trajectory, you just program a set of points along a single axis, the axis of the molecule, to it's a set of evenly spaced points along the axis of the molecule that just stretch it in a straight line in space.
How is that different from SMD?
The collective variable is different because the minimum, because of the shape of the system, I think, so this is kind of postulating, the shape of the system is this helix. And you can imagine that if you were to take a spring in real life and try and pull it out, there are going to be, it's not just going to want to uniformly pull out along a single axis. There are certain constraints in the system that mean that it might want to sort of kink up and wiggle as you're pulling it. So I think that this is why the collective variable based approach differs from the linear approach, because you're not finding this linear pathway. So the collective variable based approach is only interested in that distance. You attach a restraint to the distance between the two ends of the molecule and leave the thing to sort of rotate and oscillate freely in space. And so that allows it to do these sort of kinks and wobbles without being constrained. Whereas in the linear case, we're assuming that it pulls exactly along a line, which it doesn't. And that's reflected in the iMD case as well. Because the iMD trajectory wasn't linear. It kind of curved down at the start and then up towards the end in space.
But in the collective variable approach, it's also the collective variable is a distance at the end, right?
Yes.
So we are still pulling along a line.
You are still pulling along a line, but that line is in a different space. It's not in Cartesian space. It's in the space of the collective variable. So you have to switch to that space to calculate your forces and your where your position is going to move to next. But that's not correlated with the real space in the same way that our method is, because we're fixing one part of the system and then we create a relative path that way. In the CV version, you're creating a relative path within the molecule itself, which is a really nice way to do it, but really difficult to define for systems where you've got lots of degrees of freedom and you don't know what is going to be moving when.
And in the VR trajectory, so in it, you use VR for generating the reaction coordinate, but then you do run standard SMD, right? Without being VR.
Yes, exactly.
And can you choose, let's say, complex reaction coordinates with the VR and the simulation?
So right now, the way that I have it implemented is that you can only apply one force right now. So you can basically pull one part of your system from one part to another. But this should be very easy to generalize in a way that collective variable-based approaches aren't. Because in a collective variable-based approach, you need to specifically consider which angles, which torsions, which distances to constrain or restrain. But in the iMD approach, it's just a case of applying another harmonic potential to the group or groups of atoms, which is really easy to automate. So you don't need to explicitly apply it yourself. This is all dealt with within the class to do it. You just pass it the path that you want the thing to follow, the system that you want to use, including whichever atoms you've fixed for the simulation, as you would for preparing the iMD-VR thing. And then the indices of the atoms that you want to apply the force to. And then everything else is sort of dealt for you under the hood. So you don't have to explicitly do it.
Okay, great.
How much does it depend on how much you smooth the path?
That's a good question. It's not something that I've investigated and I need to do it more. I found that in early simulations, when you try and smooth the path to get towards a line, you approach what the linear trajectory is doing, which is good. That seems to make sense. But I've also found that it therefore will diverge from this one truth. So that's an interesting question of like how much do you need to smooth it? The parameter that I used to smooth this plot was, I think, relatively small. There wasn't too much noise in the actual trajectory itself, and I was running with a frame interval of 1. So I was seeing every simulation step in VR in real time, 30 frames per second. So the kicks weren't as big as you might get if you increase the frame rate. If you increase the frame interval and had more simulation steps per frame you're seeing.
Wait, maybe I ask you the question. Imagine you have a concerted reaction where two distances are changing at the same time. So is it possible to do it? So like pull and push two different distances at the same time?
Yeah, I mean, in principle, there's no reason that you wouldn't be able to extend it to that point. And I think that that's where I'd like to take this method. I mean, this is what I've implemented so far is like a prototype for this approach of doing an iMD-VR SMD workflow to calculate these things. And so right now it's only applying a single force to the system, but there's no reason that you couldn't pass it a list of forces or a list of paths to follow and have those paths coordinated with each other so that halfway through this one, the next one starts, or, you know, in Mohammed's case, if you're trying to enter the molecule, you know, that's something that should, there's nothing about the theoretical underpinnings of the method that prevents that. So, yeah.
Okay, great. Okay, so let's thank Harry again. Thank you. First, now we go with the last one. Last one from Aisa. So you are doing the PhD, right? With all from Santiago. You are in the first year, you said. And you will show a specific case of this bias, RMD simulations, right?
No, we are another stuff, more like pure MD results.
Pure and zero.
Yes.
Perfect. Mhm. Is the okay, great. Okay, now we increase a bit the temperature, right? Because it's hot. Or it's okay. It's okay? Okay, so.
Yeah. So you cold.
I don't know. I need or something color. So that's one degree. Okay. Sorry.
Hi everyone, my name is Glowacki. I'm doing a first year PhD at the IRL lab. And today my talk will be about an iMD-VR case study that is the synthetic receptor for glucose gluat. So the outline of my speech will be an intro, well, not an introduction, but we'll talk about a little bit more of our software Nanoware and going more in details about how the interaction works and what kind of data can be analyzed from those orbit trajectories. Then I will do an overview of this system that I mentioned before. It's a microreceptor that shows high strong binding affinity with glucose but relatively lower with other sugars. So we are collaborating right now with a team in Bristol that is currently investigating the reason of this different binding affinity. And from my perspective, my research question was if with pure iMD-VR trajectories was possible to detect, and if this tool was possible to detect the differences among the sugars binding to the complex. So just mention my background. I studied from Italy and I studied their physics at the University of Perugia and applied physics then as master at the University of Bologna. And my PhD that I'm doing at CITUS, that is in Santiago de Compostela, the CITUS Research Center, and my research in this case would be apply iMD-VR as a tool to investigate the protein system. So let's start with iMD-VR. So our software is called Nanoware, and it leads you the possibility to run real-time molecular simulation and interact with. In this case, I'm showing two examples. On the right, I'm actually interacting with the Glowacki system, introducing the sugar glucose inside the binding cavity, while on the left, there is a protein system, an enzyme called chorisapeptate, with its substrate chorismate. And well, Nanoware gives you the possibility to change the visualization and the render. So in this case, we see on the right the licorice, while on the left, the protein system is rendered as a cartoon. And as I mentioned, it gives you, you can record the trajectory and then take some data. But before going to that, I want to describe a little more how the interaction works. So basically, the user can decide which kind of force wants to apply to the system with the controller. So you can decide between among Gaussian, harmonic force, constant force. The first two will also depend on distance that your controller has between the particle you are interacting and so distance of the control. And as you can see, then you have the formula of the potential energy that you are interacting into the system. So this is the way that you can interact with your molecules. You can interact with a single molecule or with a center of mass of a certain group. Then you can record the trajectory, and then with the MD analysis, that is a well-known famous tool to analyze the trajectories, you can obtain some profiles. For example, in this case, I'm showing this, say, an example of a trajectory, the potential energy of the system during the simulation time, the kinetic energy, and then the user energy that was introduced by the user. Then you can also analyze what is the system force, that will be the internal force of the particle to the simulation. Then you can visualize the user force, so the one you have applied, and then the total force is will be asked over the two. Then right now we can also watch the work done, cumulative work done during the trajectory. The work done will be a discrete summation over the scalar product of the force that I applied to a certain particle and the displacement of this particle between two time steps. In this case, I was stretching the famous analysis system that Ari was illustrating before. One origin was restrained, one was restrained at the origin, and the other one I was manually stretching it along the axis of the end-to-end distance. So you can see the graph here of one example of this trajectory. On the left, you will see the cumulative work done, and on the right, I extracted the end-to-end distance of the diagram while I was stretching. So this is data that is currently that you can analyze, and then with the analysis you can start whatever other information you want. Since on the file, the trajectory file, we have the frames, so the position of the particles, we have the velocity of the particles during the simulation, the forces, and the one that I mentioned before. So now let's move into Glowacki, since one of those I did also the specification of things, since one of these quantitative elements I used it to analyze the trajectory I produced with this system. So this glucose microreceptor is a glucose binding exoret template. So it's a synthetic receptor that was synthesized by the Anthony Davis group at the university, at the School of Chemistry of the University of Bristol. And it was really surprising how much it was binding well with glucose rather than with other sugar. In fact, later it was also, there are also papers that published is theoretical application of this system for diabetes treatment, because through this system was then attached to an insulin receptor, and it was basically activating and disactivating the receptor, concerning the concentration of glucose inside the blood. So the structure of this system is quite simple. It's a bicyclic cage. On the top and bottom, there are two aromatic templates, while on the arms, there are three spacers that contains in total 6 urea groups. So the interesting fact is that, then apart from that, there is the main part of the structure. There are also attached to the arms 3 solubilizing groups in order to maintain the water solubility of the lower of compounds. While on the right, you see the image of the structure that we will use to the actual molecular section. So the aromatic rings on the top on the floor shows they are hydrophobic, while on the arms be hydrophilic, and this is an important fact for what explains why it binds so well with sugar, because it was actually its design was designed in a way to maximize the number of hydrogen bonds between the microreceptor and the glucose, so that the arms, the urea groups, will form hydrogen bonds with the polar groups OH group of the glucose, while the GH group, which are hydrophobic, will form a GHP interaction with the aromatics. And in fact, in this paper of 2019, they also done measurement for the association constant with other sugar, with the glucose and other sugars. And as you can see, the strongest affinity was shown by glucose because we have a binding constant of 80,300 inverse molar, while then, for example, it was interesting the fact that minor conformation differences between from the glucose structure will decrease the binding by a factor of 100. For example, this is galactose, which has an inversion of the OH nucleation position. And I also report fructose because instead it was among the one that showed the lowest binding with the microreceptor. So what I wanted to investigate if with iMD-VR, trajectory from iMD-VR, it is possible to detect these differences between the glucose, galactose, and fructose binding to the complex. So my procedure was this one. So these are explicit solvent simulation. In this case, the water molecules are hidden, so we cannot see them. After equilibrating the molecular simulation, I then started iMD-VR, in which I was applying a spring force to the sugar center of mass and dragging it out. This is just an example that a video that I made for you in order to understand better what I was doing. So I have decided to use this I spring constant because I was inspired by the SMD procedure that is the one, for example, used in the paper of 2004. So in order to have better, let's say, follow a more strict methodology. And in the end, I've run more or less, in this case, there are preliminary results. So I run 10 trajectories for each sugar, in which I was unbinding every time the sugar from the cavity to the outside from the cavity. And from those results, in this case, I'm showing the cumulative work done over the trajectory that I ran. These are pretty quick trajectories because I was inside for 20 seconds, 30 seconds, just to gather some initial result. But it's still already possible to see that there is a trend among the sugar. If you look at the final values of the cumulative work done, glucose has the highest values, but followed them by galactose and followed them by fructose. But I wanted a better method to compare them because in this case, as Ari mentioned, doing iMD, it's basically very difficult to recover any time the same trajectory that you have run in another one. That can be an issue for some reason, but for others, maybe it's like strength. So in order to then compare all the work values, what they've done is use a definition of a reaction coordinate. In this case, the reaction coordinate is defined as the center of mass of the sugar that is in purple, for example, this is the glucose compound, and the center of mass, the center of the cavity of the glucose receptor, that in this case was defined as the center of mass between the carbons on the top and bottom group. So on the left, you see a graph on one trajectory in which you can see during the simulation time the reaction coordinate that is in green in Armstrong, while in blue, light blue, you can see the work done. So to then compare all the trajectories and final work values, I decided to pick a target for the reaction coordinate, so reaction coordinate value, and then see the distribution of the work done at that reaction coordinate, so that it means that all the sugar are at the same distance from the center of the cavity. And so collecting this data for all the trajectory, this is a final normalized histograms that shows the different final work value for what concerns glucose, galactose, and fructose. It is very noticeable that they are pretty defined different range between the maximum and minimum work values for all the different sugars. And this is pretty promising, considering that iMD trajectories were done pretty quickly. I would say that we were in the order of the 7 picosecond. And yeah, so this actually confirmed the experimental result that you need, since it's the strongest bound, you will need more force to extract the glucose, and then followed by galactose, and then finally fructose, because it's the less, it has the less affinity. So the reason would be, possible reasons would be that you need higher force, since there is the glucose binds with a number high of hydrogen bond, while then followed by galactose as well, and fructose. This is something that as well, and notice the Bristol group during a molecular simulation, they have already, like they haven't already published they're still currently working on that. But yeah, this is pretty promising. So my, let's say, conclusion and plan for what concerns this site. The conclusion is that iMD-VR can detect these atomistic differences between the complexes, which it's very an important result. So it confirms also the potential of tool of this for research, just as iMD-VR approach when you want to compare, for example, as in this case, different complexes that are bonded together. So the idea would be then to tune better the parameter in order then to gather more results that confirm the preliminary one that I just shown. And then as further step, the idea is also to apply the methods that, for example, Harry mentioned, or another one that was mentioned by Dave before, to trajectory of the sugar unbounding from the complex. And ideally, an intermediate step will be find a way to define which trajectory will be the most reliable in order to do this kind of result. So this is it, and thank you.
Thank you, Ludovica. Questions for Ludovica? You know the movie where you're pulling the sugar out of the.
This is not a trajectory that I use to say that,
But the glucose, the sugar receptor has there's three different entrances, right?
Yeah.
Does it matter which one it goes through?
let's say from a matter of symmetricity of the complex, so the macroreceptor no, because it's symmetrical. When you are then considering instead the position orientation of the sugar, yes, because in fact, in this case, for example, I was comparing two different exits for galactose, and for example, the 3 or the lower, I was going out from another one respect to the other than instead of another. So I noticed that there is actually
The orientation of sugar and it goes in and out of the cavities is very important.
yes. This is something that can be factored.
Well, and you figure out what does this help you understand what the best orientation is?
Probably would be the one that will lead to less work value and less less.
Yeah, but can you see that from the data?
No, right now, no. This is like just preliminary data.
Okay.
For now, what I can see, for example, for the case of galactose, yeah, you can see that since I know that the last 4 are from a different cavity exit, probably that will be the one that it's more probable since it was easier writing it out.
Right.
If you just consider the work.
Yeah, welcome. This is a very general question. When you see this cavity, I was wondering if it's possible not only to visualize the cavity, but also some properties on top of that. For instance, the electrostatic potential, the density, just to be able to visualize better.
Inside the application, not the software,
Yeah.
you mean? No, this is not, the electrostatic like potential.
It's it'd be nice to see which is the best orientation.
Yeah, no, this is something that I was thinking about to introduce this possibility because I find it interesting as well. And yeah,
How many, not trajectories, how many cases did you explore to general?
in general, we actually for now this one because first, yeah, this couple of months I was focused on this one. For now, this is the first case that I would say.
It feels like you want to, like Boltzmann, you need to average these somehow. Right? It feels like these need to be like Boltzmann weighted. I think we need to think
But
about
that, yeah.
average these.
I was doing that in order to have a estimation of free energy. And that was also, but since we are not recording every time the same path, doing a PMF kind of difficult because we don't have the same reaction pattern.
Yes, but what you did, and go to the histogram that you showed.
Okay.
Yeah, go back, sorry, back on. I mean, what you did there is you took all the trajectories and projected them into some low dimensional space, which is this coordinate you've defined. And then you line them up that way. I mean, you could do that with those other.
I have, I can show you I have some results.
And then
I
you
need
could-
to go-
And then you could Boltzmann weight them or something. And that would just be interesting to see what that looks like.
I was checking different free energy estimator in order to do that. I can show you.
Yeah, and so why? Yeah, no, I think that's fine.
Indeed, I was wondering, if you go back please to the different profiles. So I mean, the energy is quite different, right, from one trajectory to the other. So
Yeah, but it's kind of no.
yeah, but then like in a steering ND, you need to run sometimes hundreds of simulations to
Yeah.
converse the results. Here is also the case, right? So you need, how many simulations you have to run?
In this case, I've just run 10, 2 for each sugar.
But it's not converged yet, right? So do you check the convergence of the profile?
Of the free energy profile? Or-
Yeah, or the work profile.
Uh,
Because I think it's probably far to be converged, right, with 10 trajectories on.
Okay, yeah, no, for sure. Once you get more data, that is also my idea to get more data in order to assess in conversions.
Well, I guess we need to, but the important question that Juan was asking is how would you even know if you were? So, I mean, you need a metric for whether you're converged or not. So, you're never converged in electric simulation ever, never. Even if you do 1 million simulations, you're not converged. If you do 10, you're not converged. The question is how big are the fluctuations? So you need a statistical way of saying what your fluctuations are at a given number of trajectories. And what you should see is that as you do more iMD-VR sampling, that statistical measure gets smaller. And then you have an answer to the question that Juan had just asked him, which is, are you converged? And you say, no, I'm never converged because I'm doing molecular simulation and nothing is ever converged. But I know that my variance is this.
I'm sure. So,
Right? And so, and then you can have a metric what your variance is as you use the number of iMD-VR samplings. And that's really important because that tells you what your error is, right? Right? Nothing's ever converged. Usually people think if something's fluctuating within a kilocalorie per mole, it's converged or something. It's still not. So yeah, I think coming up with a statistical way of defining the variance is important.
Okay, great. So I think we can stop here. Okay, first let's thank Ludovica and also the rest of the group. Okay, so now we can go to our environment and to leave the stuff there if you want, and we can organize a bit the time for tomorrow for the demonstrations. And then we go together for lunch. Unfortunately, we only have a reservation at 3 because it was full, completely full the restaurant, so we have to wait a minute for lunch. Spanish time. And then after lunch, if you want, we can go to the rooms and see how they are, and if you have enough space for the people and so on. Okay, so let's then go to our part.
Where should we go? Should we go to rooms before lunch maybe?
Or with one we can visit, but the other there is a guy with the key and he will go at 4. But we can visit the first. Yes. Visiting our department. So I will stop the broadcast.
Did you guys all think about? Just showing us path. It's something. But
Intenta no basar.
since we need a innovative